Good evening to our next CPS talk. It's my pleasure to welcome and introduce Dr. Suruchi Roychoudhry, who's coming to us from the Center for Plant Studies, another CPS at the University of Leeds. After studying for a bachelor's in biotechnology in India and then a master's in molecular biology from Sussex, she first moved to Leeds to do her PhD together with Professor Stefan Kropinski, where she first started working on Auxin before moving to the University of Chicago to do postdoctoral work in the Greenberg group. She was then identified as a co-investigator on a grant together with Professor Kropinski, rejoining Leeds, where she's now a senior research fellow. She's going to be telling us about her work on Auxin and how it relates to how plants sense up and down. So thank you so much for joining us.
Thanks very much. And yeah, it's my pleasure to be here today and talk about some of my work during my PhD and postdoc. It's always nice to be able to travel and do these things in person now as well. Can I just check that everyone can hear me? Yeah, brilliant, perfect, cool. Okay, so essentially I'm a plant developmental biologist, and our work has essentially focused on trying to understand the mechanisms that regulate plant architecture, in particular a crucial aspect of plant architecture, and that's growth angle. And how we really came to be interested in this topic was a train journey that my supervisor took 10 years ago, where he looked out of the window in the winter and he saw lots of trees, obviously bare trees because it was winter, and he could see their branches. And he noticed that all of these trees have different shaped branches. So if you look at the slide there, is that, you got a pointer? No, okay, that's usable. If you look at the slide up there, if you look at the trees on the top, you can see that it's often the shape of the branches that tell us about the sort of tree it is. Here, the second one is hormone, that one's mage, and then at the end you've got American speaker. So all of these trees, they have very distinct architectures, and that's of course a crucial adaptive trait as well. Now below ground, which is obviously something you can't really see, plant roots have different shapes as well. So our plant architectures, or root architectures, can range from these quite shallow angles here down to these quite steep rooting angles shown at the end of the slide. And this is something that was modeled by the Jonathan Lynch lab, again back in 2013. So why are these architectures important? So for the purpose of today's talk, I'm going to focus below ground. So I'm going to talk mainly about root work. But essentially, the shape of the root system below ground really enables the adaptive capture of nutrients below the soil. So essentially, the deeper you go, the more effect the soil strata and structure has on the uptake of nutrients and minerals. So if you look at this sort of diagram at the top, really, you find that as there's higher inputs of water and fertilizer towards the top of the soil. So, all crops, these days we all add phosphates and nitrates as fertilizers to crops to grow. And essentially, those roots will remain near the upper layers of the soil, and they're not limiting. So all plant roots can access, water and nitrates at the upper layers. The deeper you go down into the soil, the more limiting these nutrients become. So towards the middle layer, then soil really becomes an existing resource, and essentially you're limited by the amount of nutrients that are available in that soil. And even further down, those nutrients become even more limiting. So the deeper root systems can grow, the more of an adaptive fitness they have to try to basically capture these nutrients. And so there's a distinct advantage to growing roots that are essentially deeper and are able to penetrate the soil. Now, interestingly, the availability of nutrients also itself has an effect on root architecture, right? So this is some data from a few years ago, where essentially we grew some Arabidopsis plants. So Arabidopsis is a small model plant. It's similar to Drosophila in the plant world. So essentially it's a model plant. It's got a very quick life cycle. You can do lots of tissue culture with it. It goes very quickly. So essentially when you grow Arabidopsis roots in a growth medium that's supplemented with phosphate, the roots are quite happy. They grow at sort of nice angles. When you remove the phosphate, then these roots start to grow really vertically. And that's just represented in one graph down there. So essentially, the amount of nutrient available to a plant also has an effect on root architecture and particular root growth angles. Thank you. Brilliant. Thank you very much. This is what I needed. Fabulous. So just to really cement that and stress on the importance of root architecture here, this article came out again 10 years ago, again from the Journal of Textile. And what they really did was a bit of modeling to sort of really highlight the fact that steeper, deeper root systems are beneficial to crops. And that is because ideal architecture would consist of few roots that can really penetrate the soil. These roots would be thick in a sort of ideal environment, and then really be able to sort of take up all the water and nutrients that would be below the soil. And this was a study that was validated, surprisingly, not until last December. So 10 years later, the same group then has done basically some schematic modeling representation of crops, and this is in corn plants. And so what they essentially found was that in no matter what the environmental conditions were, under simulated drought conditions, so where water was limited, where plant roots grew deeper, so for example, where you had variations in architecture that look like this, or like this, all across the world, you had increased biomass above ground. So that means plants were making more corn, essentially, per amount of water. And they were also better able to sequester carbon in the soil. So all of this is beneficial from a net zero point of view. It's all beneficial from a sustainable agriculture point of view, because that is really one of the hallmarks of net zero goals going forward here. So what did we know about these angles that these roots grow at? And indeed shoots, really. So not much 10 years ago is the answer. This concept of growth angle was defined as something called the gravitropic setpoint angle. And that was a concept given by two researchers back in 1995. And they essentially defined a gravitropic setpoint angle as the angle with respect to gravity at which a plant organism is maintained with the gravity. So rather simple. And so in this context, essentially, a shoot or a plant object that grew vertically upward had a GSA, or a setpoint angle of 180 degrees. One that grew downward had a GSA of 0 degrees. Then all of these lateral obliques, lateral shoots or lateral roots had angles that ranged between these two extremes. So I've shown you here a picture of an Arabidopsis root system. And crucially, the definition of a GSA is an angle that's maintained with respect to gravity. That means when an organ is moved away from its GSA, it very quickly comes back to its GSA. So again, we give this Arabidopsis root here, and here we've got two angles in the roots that are defined by A and B. Where the root is placed on its side, this root grows downward to come back to angle B. But interestingly, this root here, A, grows upward to come back to angle B. Now, the idea of roots growing upward is fairly, I don't know, quite revolutionary to us, right? In our heads, roots always go downward, but basically that's what happens in an experimental system. So this was captured by this movie that my colleague made again 10 years ago. And I really like to look at this. So this was a movie where a picture was taken of an Arabidopsis root system growing in tissue culture conditions. And you can see very clearly that the root is sort of starting to bend upwards. So obviously, since these angles are defined and maintained with respect to gravity, it brought us back to a fundamental question, and that is, how is gravity sensed in plant cells, and how are plants able to respond to this gravity? So essentially, gravity in roots, and here again I'll focus on Arabidopsis roots specifically, is sensed in these cells at a very typical, once again. These are called polymer cells. They have a very typical architecture. They have heavy granules. They typically sediment towards the bottom of the cell. In the root that's been vertically down. And apologies to those on Zoom, but I've got a little prop here. So this is some bits I've taken from my daughter's crafting box this morning. But if you think about this as a gravity sensing cell here, and these pom-poms here, which you can see them better like this, are the statulids. And so when a root is growing vertically downward, you can ignore the colors for now. With these pom-poms or statulids will sediment in the direction of gravity. Now, when a root is reoriented away from gravity, so for example, if it's put 90 degrees like this, you see very quickly that these statulids sediment towards the new bottom of the cell here, or these pom-poms come down here. And what that does is that triggers, again, the movement of certain proteins. Now, these proteins are membrane proteins, and they're called pin proteins. And what they do is they basically look like this when you look at the minor and what they do is very quickly polarize or move towards the bottom of the cell. So this side of the cell. So here in this diagram is shown in purple. And when the satellites come to the bottom of the cell, here these spin proteins follow it. And what they do is they push out auxin towards the bottom of the cell. Now, auxin is a plant hormone. It's a very small molecule. It stands for indole acetic acid. And it does lots and lots of things in plant development. Now, one of these is basically to mediate the gravitropic response. So here is a picture of the output of auxin signaling. So if you think about auxin essentially being the green, it's an inhibits. That's what happens. So you get auxin collecting at the bottom of the cell. And because auxin inhibits root elongation, so the cells at the bottom become shorter. The ones on top are longer, and your roots will start to bend downward according to gravity. So we knew, or we had a clue that basically auxin was involved in the regulation of gravity response. But what other things did we already know about gravity sensing and response that we could take away and start to think about in the context of oblique impact? So what we did know is that, again, but essentially, gravity sensing follows something called the sine law. And again, here, if you look at this movie, I can play that again. Yeah, here we go. You see here, this is again a movie of a group that's been based on the sine and it's responding to gravity. What the sine law said, and this was again found by Sachs back in 1892, is that the amount of bending that a root undergoes during a specific time point is proportional to the sine of the gravity. So essentially, a root that's at 90 degrees starts to bend very, very quickly. And as this angle tends to come closer to 0 degrees, the response slows down. So you see the response starts to flatten. And again, this is just a graph showing some experimental data that's plotted. This is predicted data if the gravity response followed the sine law. And you can see that the curves overlap very, very quickly. And so essentially that suggests that the amount of auxin that's coming out at the bottom of the cell here, that's basically proportional to the angles at which the roots are at any given time. So could we take this and start to think about all of this in the context of setpoint angles? So what else could we sort of learn from this? So one thing we found very early on was that auxin itself could regulate growth angles. So we had this clue first when we looked at the output of auxin signaling in root systems. So we have here some roots that are fairly shallow, so this is quite small. We have here some roots that are quite old and they're fairly vertical. And so what we can do is we can use a sensor again to tell us about the amount of auxin in a root at any particular time. And this sensor here is called related roots. And here this is an inverse proportionality sensor. So the more auxin there is, the less signal there is. The less auxin there is, the brighter the signal is. It's inverse proportionality. So you can see in older lateral roots here that are quite vertical already. We have more auxin because the signal is quite low. And in young roots are relatively horizontal. We have quite a lot of signal here. And we have lows. So that essentially means or suggested to us early on that auxin itself could make roots grow more vertically. And this was validated in an experimental finding where if we took our roots, we treated them with auxin, we could make roots grow more vertically. So they were steeper. So all of that started to give us some clues about this mechanism. And so at the time, to try and understand what could be different about lateral organs and from the primary axis, we came up with two possible hypotheses. These were fairly passing organs in the sense they were simplistic and they were minimal. And I'll try and talk you through them. So the first thing that we hypothesized was that if these satellites in the primary axis are triggering gravitropic growth by interacting with some receptors at the bottom, triggering growth. One possibility could be that in a lateral root that's growing at an angle, these satellites are at the corner here, and perhaps the receptors that they're interacting with are in this corner here as well. So that was one hypothesis. Or the other hypothesis was that there was nothing changed about the receptors. The roots, when they were at this angle, were trying to grow downward, but there was something else that was pushing them upwards and stopping them moving downwards. So they were the two sort of, balanced or equivalent hypotheses that we came up with 10 years ago. But how to test this? To do this, again, we used an instrument that was first used in the 1800s, and this is called a clinostat. Now, what this clinostat does is basically it rotates down very, very slowly. So you can set the speed of rotation. So the speed of rotation can range from 1 revolution per minute to 4 revolutions per hour, and a whole range in between. But essentially what it does is, it's because it's rotating continuously very slowly, you're removing the plant's reference to gravity. So the plants can't sense gravity anymore because you're constantly changing the orientation of gravity. So you're totally just removing that reference to gravity. And so what we did is we put both roots and shoots on the clinostat to see what kind of growth patterns we got. And interestingly, our shoots started to grow outward and our roots started to grow outward. And so we sort of really came to the conclusion that this was the likely scenario, in that where the vertical axis was growing downward, but in lateral orbits, we had two balanced fluxes here shown in green and red. The green flux of the green Auxin flux was constantly trying to push the root downward, but then there was an antagonistic red flux that was trying to push it upward. Now, in a clinostat or a clino-rotated situation, we essentially remove the green bit, and so we only have the red bit. That is why our roots were bending upward and our shoots are bending downward. So if you just think about this being maintained at an angle, sort of red and green, when you start to rotate, you remove the green, so the roots start to go up because you only have the red. So in the same context, if you look at my box here, that's really where the red and green come in. So there you put your antagonistic flux, your gravitational flux, you remove the green. So that was all well and good. And we can sort of capture some of these dynamics in this very classic experiment here, which I'm sure we have all done in school sometime, where you basically take a stem of a plant and you just put it on its side, and you see that the stem starts to grow upwards very much slowly. But what I'd like to draw your attention to is this secondary branch here. So if we just play this movie again, from the beginning, when we put the plant on its side, for a few minutes, we essentially remove the reference to gravity. The plant's not set before the gravity sensing mechanism kicks. And you can see in those few minutes, this stem bends completely backwards. So it's like a clinostat situation where you kind of remove the reference to gravity briefly, and your stem's now bent. One more time, hopefully we'll catch it. Yeah. You see the stem bends quite far back, exactly like the one clinostat. Then eventually starts to connect itself, starts to sense gravity again. The primary is going upward as you expect. This also starts to sense gravity, comes down upwards eventually. So that's quite a cool validation, we thought anyway, of our hypothesis then. And that's just a diagram just showing you, just tracking the movement of that branch there, and the angle plotted which happens to die. So all of this, really, we could apply in the context of our sine law. So essentially, again, if you remember what I said about the sine law, where the gravity response is proportional to the angle of stimulation, you can see in a root that's growing sort of vertically downward, or sort of these angles, we put a balance between the green flux and the red flux as well. So these roots are maintaining that oblique angle. Now, in a root that's bent upwards, away above its GSA, you've got a stronger green flux here at the bottom. So you've got more Auxin being driven down. That's why it is bending down. And exactly the opposite is the case when a root's bent below. So in this case, when it's bent below, it's below angle. This green flux gets according to the sine law. Because the red flux is now bigger by comparison, your root is moving back up. So this was what the model would have predicted. Could we validate this using our Auxin reporters as well? And the answer is yes. So again, if you remember, this is the way to Venus again, so it's inversely proportional to the first signal. So at Gravitropic Setpoint Angles here, we've got a nice balanced signal on both sides. There's no asymmetry really. This is equal amount of signal on both the upper and the lower side of the root. If we take the root and put it above its GSA, you will have an expected increase in Auxin at the bottom. And so because you have the expected increase in Auxin here, you have a reduction in signal because related increases versus both. All of that's just been captured in that graph here, where we quantify the fluorescence for half the and exactly the opposite happens in this situation here, where you're expecting to have very more oxygen at the top than one. And so your signal is slightly weaker at the top, slightly stronger signal. And just to reiterate, this is more difficult to capture than this, because here you put a reduction in signal on the side rather than the other. But if you quantify the fluorescence, the upper and lower side experiment, you can actually see that yes, that is a significant amount of signal that we see in all of these situations. So all of those findings led us to propose this model. Again, this is back in 2013, where we basically said that roots and shoots indeed, and we are mainly focused on maintain these oblique angles through Auxin acting to maintain what we call gravitropic Auxin flux, as well as an anti-gravitropic Auxin flux in red. And that was really the first model of GSA control back in 2013. So we also said, and I haven't talked much about this bit here today, but Auxin itself through Auxin signaling can reduce. And that was validated by our simple experiments where we treat our list with Auxin, they become more. So essentially, Auxin's acting is very, very short. And you get more through signaling components as well. So all of that was fine, but what were the molecular players behind this? And so we come back to our old friends, the proteins here, which act to push Auxin out of the cell. So according to that sort of gravitropic response and sensing model, what we know is if we have a primary root here, if we have a root that's going vertically downward, and if we put it at an angle here like this, essentially our statoliths would come to the bottom of the cell. If we look at it like this, we would have an expected increase in pin proteins here at the bottom of the cell here. And that would then push Auxin out and through bend downwards. And if you do that with the primary root, you put it at an angle. That's exactly what you see. You see an increase in pins on the side here. And if you make a quantification of upper to lower side fluorescence, the primary axis, you see that actually those ratios are lower than 1. And this is what we're doing now, which is exactly what it is. But in a lateral root that's already growing at an angle, that's not what you see. You quantify the amount of fluorescence in the lateral root that's going the upper and lower side. You find that these ratios are very close to 1. So obviously, there's something different about the pins in the lateral root, even though they're the same protein, they're behaving differently, right? Even though they're not responding to gravity in the same way. And what's making them do that? That was the next question that we set out to answer. So we did some quite clever microscopy again in our own words, at least, where we did something called flip assays. This essentially is where our end comes. So we used a vertical confocal microscope to look at our cells in this orientation. So a microscope was here, this was our eyepiece, we could visualize the cells like this here. And then what we did is we flipped the cells. We put them here, the top, and looked from the top of the cell to see what was happening to the pins on the upper and the lower side. And so when we did that, and this is the same cell of both pins, pin 3 and pin 7. You can see here essentially that the 0 max, right after we flipped the roots this way, there was a bit more signal to the bottom. It was almost on both sides. But then after half an hour of having the roots like this, we lost signal from here, but we still had signal on the red side. And that continued at 60 minutes. And then finally, after 2 hours, signal then kind of became dark and balanced on both sides as well. And that was the case for both pin 3 and pin 7. You can see this ratio upper to lower fluorescence is higher than 1, particularly in 40 minutes. Then gradually comes back down to 1. So the signal balances out. So that essentially told us that the pins on the red, the upper side here, were hanging around on this membrane for longer than they were on the lower side. So there was something about them that was making them more sticky on that upper membrane. And what could that be? So we started to explore post-translational modifications here. And looking at the literature, one of the post-translational modifications that we use, so just to highlight, this is very biological, but essentially proteins behave differently with sort of chemical modifications on their backbones. So they might have tags on them. So protein has an amino acid sequence, and then they can have different tags on them. One of the most common tags that they could have is phosphate groups, essentially. And that does a lot to the behavior of the protein. It changes how proteins behave in cells. So this is a simplistic diagram of what these spin proteins look like. So they have these transmembrane domains. If you remember, I told you that we go to the cell membrane here. So these bits here go to the membrane, bits. And then this straight bit here, so it hangs around inside the cells. But these bits here, they have a lot of serine amino acids, and these amino acids, serine groups can have phosphorylation groups on them. And what we knew from the literature was that essentially phosphorylation does something to the activity and the downstream of proteins as well. So we started to look at the effect of phosphorylation in lateral groups, primary endpoints as well. And we also knew a few sort of players in that pathway. So we have proteins called phosphatases and kinases, again, if you're getting that essentially act on these proteins. So we started to look at their localization and their behavior in the cell as well. So just to cut a long story short here, what we found is that when we have our Arabidopsis plants expressing sort of a normal, a wild type version of this SPIN3 protein, the roots grow, well, as you would expect, straight. If we engineer this protein using a technique called cyclic mutations and add some more phosphate groups here, our roots start to get shallow. And again, if we do another set of engineering, and if we remove these phosphate groups here, then our roots tend to get more motivated. That's just quantifying surroundings. So your growth angles on average, they're shallow when you have these extra phosphate groups, and they're more motivated when you use the phosphate groups there as well. And so that gave us a pretty good idea to say that phosphorylation was actually playing a role in regulation of growth angle and possibly pin polarity. But was it doing something to the localization of the pins as well? And again, the answer is yes, really. So I don't think my questions have yes answers here because the nature of the talk. But anyway, again, to cut a long story short, we found that actually at roots that had a wild type-ish, so normal version of the protein, then loose groups, so very angles, you had about the same amount of pin3 on both sides. And this was the same thing as wild type. So you can see the ratio is very small. When we have lots of phosphate groups on the protein, the roots move shallow angles. You can see that actually we have more pin on the other side. That's true. So the ratio is much higher than that. And exactly the opposite. So when you had roots going over to the essentially you had more pin including one, and that ratio is lower than 1. So we could conclude here that basically phosphorylation was affecting pin polarity. But again, through what mechanism, through what protein? Were there some other players involved that were doing this? And again, the answer was yes, there was another one. And there was a phosphatase protein that we identified that played a key role in regulating the phosphorylation status of these spin proteins. So this is a phosphatase protein. Phosphatase is a protein that removes phosphate groups essentially from other proteins. So this was called the RCN1 protein. And what we did is we basically found an Arabidopsis mutant that doesn't express RCN1. So when you have a protein that, a plant, transgenic plant, that's not expressing the particular gene, it's a loss of function mutant. And so we found that a loss of function mutant of RCN1 has shallow angles. And that's shown in this graph. You see it's a shallow angles. Now, if we take this phosphatase and if we express it specifically in these spin columns, that is a few cells here. So you do that again using protein machine. So if we express this RCN1 only in these lactic cells here, we are finding that we can actually make out this more. And so the expression of this phosphatase solely in the lips is sufficient to engineer plant roots to be more vertical as well. And how does it do that? The answer is it does it through regulating pin polarity, of course. So again, if we come back to our pin proteins, our friends, and we look at the targeting of these pins in this AR structural plant that's expressing RCN1 only in the what we see is that the wild type version, the control version, normal version, the average ratio is very close to 1:3. And from one sample, like if we look at the other thing, let's say already we see that actually it was done. It's more increasing towards the one. So this is some of the but this is not the case for the other things. So other closely related in 7 actually is unaffected by RCN1. So using all of these findings, we decided to test whether auxin itself has an effect on RCN1 protein level or expression. And so what we did is we took transgenic Arabidopsis plants again, expressing fluorescent RCN1 GFP. So this is RCN1 protein back to a GFP marker. So we can actually see the protein cells. And if we treat these roots with auxin, and specifically the columnar, which is a root here, we found that actually the amount of RCN1 increases. So the greater signal means more protein here. So you actually see that we've got much more signal out here. So basically, auxin itself is directly increasing the amount of RCN1 in the cells. And so if we take all of those findings together, we can now come up with basically a model that's version 2, device 2. So essentially what we basically said is that auxin is regulating these two classes. You've got a red and Gravitropic Setpoint, and then a green Gravitropic class that's anti-hormone. Auxin then is inducing RCN1, which can basically change the form of PIN3. So you can change the balance between phosphor and phosphorylated PIN3. You can have increased PIN3 targeting towards the bottom of the cell here when you treat auxin. And that's how you're going to rank. These findings are currently interested and hopefully should publish sometime soon. So that's essentially where that story is up to. If we've got 5 minutes, I'd like to talk about last section of the talk, which is a bit more to do with gene editing and has some more real world applications. But I'll go through that fairly quickly. Exciting. Right, okay. So we had a pretty good idea of how auxin was regulating GSAs, but in our search to do research with more impact, essentially, we decided to also look for novel regulators of root growth angle. So look for genes or proteins that were affecting root growth angle, either independently or independently. And as part of this, a few years ago, one of my colleagues or 2 colleagues did something called an EMS mutagenesis screen. So this is a screen where essentially you can take Arabidopsis seeds and treat them with a chemical mutagen. So one that essentially would cause cancer to humans. So you're basically you're causing lots and lots of random mutations in your plant genome. And you don't know where these mutations are. They happen completely random. And so what my colleagues did was to get a bunch of these mutagenized seeds and then test them for root growth angle. So they plated them on these tissue culture plates and they looked at the angles. And so they looked at thousands of plants, got a long story short, over a few years. And they found this one mutant that they called 81.1, had quite vertical growth angles here. And that's just a comparison for a normal plant. So that was exciting. And the idea was to see how this mutation was essentially causing vertical growth angles. And so my colleague sequenced the gene and found the mutation behind it. Now, I can't, unfortunately, tell you the name of the protein because it's been licensed patented. So we're not going to take my word for that, unfortunately. But essentially, what they found was that the mutant was essentially a single amino acid change in this 81.1 line. And that single amino acid change led from an arginine amino acid, which looks like this. You guys are chemists, so you understand this protein structure. Basically to a lysine. So you lose one side chain that goes in this particular position. So that single mutation is sufficient to confer these excessive leaf vertical root growth angles here. So to get an idea of how this protein could be affecting root growth angles here, we did the opposite. So we took some more plants that had lost this protein, so loss of function agent. And we saw that actually they had shallow root. So that gave us a pretty good clue that this protein and this gene was definitely regulated. And then we also looked at PIN3 polarity, again, in these loss of function agents. And we found that actually these roots are growing more shallow here. there's an increased membrane fluorescence to the other sides. There's no utilizing to the other side here in these mutant proteins. So all of that was very convincing to us, and we realized that actually we've got a good candidate that regulates root system architecture, and in particular root growth angle. And so what my colleague did was to essentially do some gene editing using cytogenetic mutagenesis in Arabidopsis and to verify this hypothesis. So here again, you've got your normal wild type plants. Here you've got your loss of function mutant, so the one was the shallow angles. This is the 80.2.1 mutation that you identified. What you could do is you could clone out the version of the gene from this 80.1, put it in this loss of function. You can see that now we can make this loss of function look pretty much like wild type. If you put this 80.2.1 really essentially Columbia, so wild type pattern, you get more genes. And if you engineer that same position with different amino acids, so if you remember that original substitution was not changed to lysine. If you change that lysine to any other amino acids, lysine, this is the acid, you can see that you still get more genes as well. So essentially, a single base pair amino acid change in that position is sufficient to basically give us significantly steeper roots. Now, excitingly, we found some niche lines that were bearing the same mutation here as well. So again, PhD student in our lab, Faye, she characterized the growth patterns of these niche lines. So cadenza is a variety of wheat that's quite commonly grown in wheat. And so sort of normal wild type wheat cadenza has angles and like these are roots that are grown in pouches. If you take cadenza wheat bearing this 80.1 type mutation, you can see that the roots are actually moving. They're actually smaller as well, shorter. And that's just data that's qualified here in this graph. So this is wild type wheat along the roots. And you can see at every point after emergence, this 80.2 mutation makes the roots grow more here. So preliminary data from soil trials as well showed that actually these root angles remain more vertical in soil as well. And these images are taken using an X-ray CT thermograph. So you have roots growing in pots in soil and you can take it. So it's quite a good technology, but it's quite limited in terms of how easy it is. So essentially, this mutation of this gene is being licensed at the moment, and field trials are currently underway in collaboration with an ag biotech partner at the John Innes Center. So fingers crossed that that will lead to some, some exciting gene-edited crops in the future. And so I think for my final slide, really, I just want to highlight that research at Leeds, not just our research, but research, because the Center for Plant Sciences at Leeds is currently focused on at different scales. It's really biology across the scales. And I think this story is a nice example of that, because what we found is essentially a single sort of change at protein level changes how the proteins behave, which then leads to differential growth, and then actually translate into some different systems, bigger plots, and hopefully give us mechanisms going forward. Yeah, and with that, I'd like to thank all of the people that did this work. There were many, many more reasons that came to mind when I was coming down the train. Stefan Kulinski has been my PhD and postdoctoral supervisor. Many PhD students behind this work, Andy Martina, Katie, and collaborators across Europe as well as the Living Trust, UKRI, and VBSRC for funding. Thank you very much for your attention. I'd be happy to take questions if there's time.
Absolutely.
Thank you very much for the talk. I was wondering for the future field tests, do you have sort of what applications are you looking for to do that?
Sorry, could you repeat, please?
For the field testing experiments, which locations do you have in mind?
Oh, yeah, that's a good question. So field trials at the moment they're UK based, because I think the variety of wheat that's being tested is cadenza, which is basically British spring wheat. But there's no reason why in the future going forward these locations could be worldwide. It depends on finding an appropriate I think, licensing partner and validation of the, so we call this step, and I'm learning all about this as we go along, because essentially I'm a developmental biologist. So all of this licensing and field trials is all quite new to me as well. But essentially, this step of the work is called technical valuation. So this technology is validated at different levels. So it would be then transferred to different wheat varieties that could be grown across the world. And then these would be tested first in controlled conditions. So in, for example, in glass houses, where you can really control, you can measure lots of parameters like rainfall, how much water the plants are getting, you can measure their yield. And then if those data look good, so that is preliminary data, then essentially it's transferred to fields essentially, and it could be all across the world. It's a very similar process to, I guess I would say, like drug development, where you have phase 1, phase 2, and phase 3 trials. It's a bit like that, except there's fewer ethical approvals because there's no people involved, but there's still a lot of genetic regulation that you need to work around, especially as because different parts of the world have different rules on what constitutes genetically modified organism versus gene edited organism as well. But that's the aim. We want to save the world. One crop at a time. Yeah.
Thank you so much for your talk. I'm a master's student doing microscopy at the moment, and I was wondering if you were going back, what advice would you give to yourself?
Specifically with regards to microscopy?
With regards to choosing your career and...
Yeah, that's a good question. I guess like it would be, don't second guess yourself, really, because I feel like a lot of the times the things I did, I wasn't confident about the choices I made, but I think they came from a place of instinct, perhaps, or knowledge. And so I was making the right decisions without necessarily knowing that. So I guess I would say, like, have faith and trust the process, trust yourself, believe in yourself, essentially, have confidence. You'll get that. And it's all right if your goals change as you go along, because you might decide that, you change your mind about what your final achievement is as you go along. And that's fine as well. Trust the process. That's what I would say.
Thank you.
I enjoyed answering that.
I've got a question. So one of your mutants, the phosphatase mutants, and you've got your patented mutant as well. So the phosphatase mutant increases makes the angle more shallow, more steeper, right?
Yeah, shallow. Yeah, phosphatase mutant.
Okay. What will happen if you combine both of these?
Yeah, good question. So my colleague,
Can you make that and then
yeah.
can you combine this?
Yeah, so essentially, if you combine the mutant that's got steep root angles with the one that's got shallow angles, you end up like an Indian. So one of my colleagues, his PhD student, he's trying to work out if there's a genetic interaction between both of these. So this gene and also the phosphatase, and so that's been the simplicity we'd obviously like it to be in terms of having stability, but we have to accept that there could be parallel into the recognition of the angle essentially. So there's been
One piece, I'm going to just...
No, it's okay. I've got my own, thank you.
Oh.
Hi, sorry. Are you trying to ask a question on Zoom? Yeah, I'm trying to get to the chat. I got a comment saying the lecture is interesting. Oh, thank you. Yeah.
I guess maybe while we're waiting to see if there is a question.
Yeah, exactly.
One question I was wondering is, at the beginning, you talked about how there's sort of the increased biomass above ground when you have the deeper, steeper roots. What you were sort of showing at the end, where you can get these mutations, how much of that biomass is because the roots are deeper and steeper versus the mutation that's causing the deeper and steeper roots is causing more growth?
Yeah, absolutely. Great question. And I guess the answer is we're not fully sure yet. And I guess it like different mutations will have different effects on. So a lot of these genes, they obviously regulate root growth angle, but they potentially regulate lots of other processes as well, especially anything related to auxin, because it says essentially we call it a master regulator of plant growth. So it does, lots of things. And so the answer is we're not really sure yet. But then I guess in the sort of grand scheme of things, does it matter, in that sense? Because at the end of the day, what we're trying to do is basically get like another concept that we use is essentially called more crop per drop, so reducing your agricultural inputs and trying to get as much yield, if not more, and that's all from a net zero sustainability point of view, for example. So for example, again, I didn't talk about this much today, but the production of nitrate-based fertilizers, you probably know more about it than I do, but it's called the Haber-Bosch process. And again, it's incredibly carbon intensive. So it will, in terms of decarbonization, it's not good, basically. So anything we can do to reduce those inputs and really try and maximize, productivity above ground is a step in the right direction as we see it. But that's a great question from certainly from a developmental biology point of view. And it's one amongst many that we still need to answer. Thank you.
We do have a question,
Oh,
which
yeah.
is how the waste phosphorus products removed in the soil.
Good question. They're not. They end up in so phosphorus is supplied in the form of phosphate fertilizers. And phosphate-based fertilizers tend to go deeper into soil layers because they water soluble, whereas nitrates tend to spread off. So essentially, they just accumulate in deeper and deeper soil layers. With nitrates, the opposite happens. So you might have heard of this concept, eutrophication, where essentially a lot of nitrates wash off from the soil and they end up in like ponds. And so you have this excessive algal bloom, algal growth. And that's because essentially, the Green Revolution back in the 1800s, really, a lot of it focused on making better shoot architecture. So having plants that were shorter and basically producing more, but a lot of the below ground traits like roots were completely ignored. So actually our roots, the plant roots as they stand, are not very good at taking up phosphates and ions, because all of the Green Revolution happened during times when there was a lot of availability of these fertilizers. And it's only now, with things like the Ukraine war as well. So for example, a lot of these fertilizers are manufactured in that part of the world. And they've obviously become limiting again as well. And so this is why there's been it's been a focus of discussion with the development of plant biologists that the second Green Revolution essentially will be below ground. So yeah, that's just ongoing discussions.
Well, thank you so much for your talk.
Pleasure.
And for answering all of our questions. We really enjoyed learning more about your work. So let's thank Dr. Roychoudhry again.
Very much.